Hi, everybody, and welcome to this week's lecture. What we're going to be covering today are going to be cell membranes as well as diffusion. So, what our learning outcomes are going to be is we're going to do a quick review of exactly what the cell membrane or plasma membrane is. We're going to go ahead and learn the importance of our concentration gradients as well as diffusion. We're going to learn about passive transports and the different types of protein transporters. And then finally, we're going to go ahead and look at active transports as well as protein pumps. So we have a lot to cover in this week's lecture. There are going to be some learning checkpoints that we're going to go through. I'm only going to answer, maybe look at one of them just for the sake of time. But again, you're always more than welcome to spend as much time as you like on those test your knowledge portions. So let's go ahead and get started. The cell membrane. Now, when it comes to the cell membrane, one of the most important things about this is that it is going to act as a barrier between the inside and the outside of the cell. So if I were to go ahead and outline all of this all around our cell, this would be our cell membrane. Again, it's providing a physical barrier between the outside and the inside. Now, if we were to go ahead and look at that as an analogy with a house, the cell membrane would be the same as the walls of our house. And again, the reason why is because just like the cell membrane, the walls of a house is meant to separate the outside environment from the inside. So again, the cell membrane is very similar to the walls of a house. And it's going to be doing that via the lipid bilayer. So remember, a lipid bilayer is comprised of a hydrophobic and a hydrophilic end. Lipid bilayer, bicycle, 1, 2, bilayer made up of 2 layers. And you can kind of see that over here. Again, you can see the head as well as the tails, and they're binding together in that bilayer section. Now, the cell membrane is going to have membrane proteins embedded into it. So it's going to be all of these kind of pieces over here. Now you can see that there are very many different shapes and sizes, and the reason why is because we have many different membrane proteins, and they're all going to perform different jobs. Some of them including transporting molecules across the membrane, cell communication, enzymatic activity, as well as anchoring cells to other cells or extracellular substrates. So now let's go ahead and take a look at some of the movement across the cell membranes. So when it comes to the movement across cell membranes, we're going to utilize something known as transporters, channels, and pumps. And this is going to help regulate movements of molecules in and outside of the cell. So if we were to go ahead and compare this again to our house, the house is going to have a door and windows that are going to allow things in and outside. And that's going to be the same idea when it comes to the cell membrane. You'll notice that the cell membrane is going to be semi-permeable, so it is going to allow certain things in and out, and some molecules are going to have free movement across the membrane. Some of them aren't going to have free movement, but more as a regulated movement across. And again, they're going to get through with our protein transporters here, protein channels, as well as our protein pumps. So again, the cell membrane is something known as semi-permeable, meaning that it's allowing some things in and some things out. Now, some molecules, again, can freely pass through without any issue. So again, they can come and go as they please. Some of those things are going to be oxygen, carbon dioxide, and water. Now, some molecules, although they're going to be very important for our cell health and life, are not going to be able to come in and out as they please. Although, again, they're going to be very important, we're going to need to utilize some of those protein channels and pumps in order to let these in. So they're going to be much more regulated. Some of those are going to be charged ions, sugars, as well as amino acids and proteins. All right, and that is going to conclude our review for our first part. So go ahead and take a couple seconds, pause this, and see if you can answer these 3 questions. All right, welcome back, and this is what you should have gotten. One of the ones I do want to kind of touch on is going to be this over here. Which of the listed molecules can freely move across the cell membrane? It should have been oxygen and carbon dioxide. Again, if we go back to this over here, you can see some of our molecules have free range to come in and out, while some of them are going to be much more regulated. Now, this is going to bring us into our next part, which is going to be known as our concentration gradients. When it comes to concentration gradients, the main thing you need to know is they're going to occur when there's a concentration of particles in high areas or higher than other areas. So that means in this case, we have an area of high concentration and an area of low concentration. So if we were to go ahead and look at an example of this, we can look at cookies being baked. Now, if you bake a batch of cookies and you take them out of the oven, they're fresh, they're going to smell very, very good. And they're going to smell exceptionally good when you're very close to them. So that means the smell is going to be high when you're very close to them. But if you were to go 1 or 2 rooms away, you're not going to be able to smell them as well because you will be in a lower concentration of that smell. So in this case, the closer you are, the more concentrated those smell molecules might be. And then the further away, the lower concentration they're going to be. So again, the main idea to get from the concentration gradient is you're going to have an area of high concentration, so an area where there's lots and lots of stuff. In this case, you can say the smell of cookies. And then there's going to be area of low concentration or much less particles. In this case, a much lower amount of that smell of those cookies. So the next thing you need to look at is going to be diffusion. Now, diffusion is going to happen when you have an area of high concentration and an area of low concentration. And what's going to happen is you're going to have random movements of those particles, and they're going to diffuse or move from an area of high to an area of low. Now, if we go ahead and look back at our example with our cookies, remember when you're really, really close to those cookies, those freshly baked cookies, they're going to smell really, really good because you have a very high concentration of that smell. And then if you move 2 rooms away, you'll have a very low concentration, so it'll smell very, very faintly. But over time, as these cookies sit out, you're going to have a movement of those smell particles diffuse into the lower concentration. And eventually, something known as equilibrium is going to happen, where there's going to be an equal distribution of all those smell molecules. And again, the reason why is because all molecules are in constant motion or that constant random motion. So they're going to randomly be moving around until we hit that equilibrium. Now, this can also happen in liquids as well. The example that I'm going to use is going to be dye or food coloring and water. Now, if you take a drop of dye and it hits the water, you'll see that there's going to be a very high concentration of color. So you'll see a very distinct color difference. You'll see the water, which will probably be clear, and then you'll see the dye color, in this case, purple, very, very concentrated. So we have high here and we have low over here. Now, over time, again, we're going to have that diffusion where this is going to diffuse or separate out. And you can see that in step 2, that the concentration level went from high to about medium because they are diffusing or moving around until they are equally distributed, again, hitting something known as equilibrium. So in this case, our dynamic equilibrium is reached when particles are uniformly distributed, meaning that they're going to be fully distributed throughout the entire solution. Now, why is this important? Why do we need to know this? And how does this apply to living organisms? Well, diffusion is going to be incredibly important when it comes to living organisms, specifically for oxygen and carbon dioxide. So if we go ahead and take a look over here, I have an example of a lung, and we have an alveoli, which is going to be these little air sacs that are going to expand with oxygen, and then you can expand them again and exhale with carbon dioxide. Now, looking at this, whenever we breathe in, we're increasing the amount of oxygen inside our alveoli. So now we have a high concentration of O2. Now, in our blood, we know that's a major transporter for oxygen as well as nutrients. So whenever we're trying to oxygenate blood, we want to get the oxygen from our lungs into that blood. So that means that we have a low concentration of oxygen right now in the blood. So our lungs have lots of oxygen, blood do not. And because of that diffusion, again, going from an area of high concentration to low concentration, that oxygen is going to diffuse or move into the blood, causing our blood to now become oxygenated. So again, we breathe in, take in a lot of oxygen, and because we have that concentration gradient, meaning we have a high concentration here and a low concentration in our blood, that oxygen is going to diffuse out of our lungs into our blood, causing our blood to be oxygenated so that it can go through the rest of our body. And the exact same thing is going to happen, but inversely for carbon dioxide. When our blood is saturated with CO2, so we have a high concentration now in our bloodstream, and we want to get rid of that. So what's going to happen is our blood is going to come by near our alveoli, and it's going to diffuse out into our lungs because we have a low concentration of carbon dioxide. So again, high concentration of CO2, it's going to come by, hit our alveoli, it's going to diffuse the CO2 into the alveoli, and then we're going to go ahead and exhale it out. So again, that's an example of diffusion in living organisms. Another example would be this cell here and water. Water is important, and water is going to be one of those molecules that can passively diffuse or move in and out of our cell. So in this case, if we have a high concentration of water on the outside and we have a low concentration on the inside, again, we are going to diffuse through, meaning that the water is going to move from that high to low until we hit that equilibrium. All right, so that's going to conclude our second part. So go ahead and take an opportunity to look at our review questions. I'll give everybody a couple seconds to pause this and give it a try. All right, and welcome back. One of the things that I do really want to stress is this right here, number 2. In a solution, substances tend to move from an area of high to low concentration. So remember, anytime you're dealing with diffusion, you have to have that concentration gradient. Remember, an area that's going to be high and an area that's going to be low. And when you're diffusing, it's always going from an area of high to an area of low until you hit equilibrium, meaning that both sides are going to have roughly around the same amount. All right, so the next part we need to look at now is going to be the different types of transport. Now, the very first one we're going to look at is going to be passive transport across the membranes. And again, we're going to do that via diffusion. So if you remember, there are certain molecules that can move freely throughout the membrane. So we don't need anything special. They can kind of come and go whenever they need to. Now, this is going to be known as our passive transport, and the reason why is because it is not going to require any energy. Because we have an area of high concentration and we have an area of low concentration, again, they naturally want to hit equilibrium. They want to be equalized. So in this case, we have a high concentration of oxygen here, low concentration here, so they're naturally going to want to move out into the cell. So again, they're going to diffuse in an area of high concentration to an area of low concentration. Now, examples of this, again, would be lung and blood. For our lungs, we're going to breathe in, take in lots of O2, meaning we have a high concentration now. And then our blood, which again wants to become oxygenated so it can bring that oxygen to its tissues, has a low concentration. And because of that, we want to hit equilibrium. The oxygen is going to naturally want to move over or diffuse into our blood. So again, moving from high to low. Same thing over here. Now, after we've breathed in and the blood has taken the oxygen has now gone into our blood, we now have a high concentration in our bloodstream. And the cells you want to get it to, whenever they have depleted the amount of oxygen they will have a low amount. So when our blood comes by, it's going to want to diffuse the oxygen it just took in into our cells. So again, going from high to low. All right, so our second type we're going to look at is going to be known as transporters. Now, we're going to be looking at things that cannot freely move across the cell membrane. The example we're going to look at is going to be glucose. Again, glucose is going to be very important. It's one of our major macromolecules. And you can see that these glucose right here cannot move into our cell. Now, what it's going to do is it's going to go ahead and use our transporters in order to allow that exchange to happen. So these are going to be right here. And these are what's going to allow the glucose to move, again, still using diffusion, area of high concentration to an area of lower concentration. So again, they're going to come in here, and then they're going to go ahead and get released into the cell. So again, glucose is going to be using more of a regulated transport. Our next type is going to be channels. Now, the major difference between our transporters and our channels are going to be its ability to open or close. So in this example, we're going to look at sodium. And again, sodium is another molecule that cannot freely move across the cell membrane. So unlike water, oxygen, things like that, they cannot freely move through. And they still require some sort of passive transport. So in this case, they're going to be utilizing our protein channels. Now, protein channels can be opened or closed, and they're still going to use diffusion, going from an area of high concentration to an area of low concentration. So whenever these open up, we have a high concentration outside the cell, and it's going to go and flood and come inside, where there's an area of low concentration. Whenever we don't need this anymore, they're going to go and close up, and then they will not be able to freely move anymore. So again, the major difference between our channels, our transporters, right here, and our channels is its ability to open and close. All right, we're going to go and look at our final one, which is going to be known as our protein pumps. Now, the major difference between protein pumps is it's going to require energy in order to operate. So the example we're going to look at is going to be our sodium and potassium. Now, sodium and potassium, again, cannot freely move across the cell membrane, so they're going to require something that's going to allow them to move. In this case, it's going to be a protein pump. Now, protein pumps are going to be the opposite of all the other types of transporters channels we looked at. All the other ones required passive, meaning we didn't require any energy. They kind of just happened when they needed to. Now, in this case, it requires ATP. Remember, that's our energy source, adenosine triphosphate. Now, what's going to happen here is protein pumps are going to work against the concentration gradient. So that means that they're going to pump in the opposite direction. So unlike diffusion, where it's going from an area of high concentration to an area of low concentration, protein pumps are going to do the opposite. They're going to move ions from low concentration to high concentration. So they're going to actively force it the other way. Naturally, it wants to go from high to low, but when it comes to protein pumps, it's going from low to high. Okay, so again, when it comes to protein pumps, we're using ATP, adenosine triphosphate, in order to take whatever we need to go against the concentration gradient. So it's going from an area of low concentration to high concentration. And again, this is going to require ATP, or our adenosine triphosphate. So this right here is going to be a table of all the different types of transports across the cell membrane. So I would definitely star this, circle this, do whatever you need to do to kind of remember this, because this is going to give us a really good example of exactly which ones are passive and active, and what the flow direction is going to be, and if they require energy. Again, diffusion right here, you can see these are examples. Oxygen, CO2 can diffuse freely. They don't require any type of protein in order to move. They're going to be moving from high to low, and they do not require any energy. And again, these are all going to be our passive, meaning it requires no ATP. Facilitated diffusion, again, glucose transporters are going to be moving from high to low as well, so they're still using diffusion, and it's not requiring any energy. Facilitated ion channels, remember they can open or close, moving from an area of high to low, and again, do not require any energy. The only one that does require is our active transport. In this case, example is sodium potassium pumps, and they're doing the opposite. They're going from low to high. So they're working against the concentration gradient, and this requires energy in order to pump it in the reverse direction. All right, and just like that, that is going to conclude our lecture on the cell membrane and the different types of pumps. So again, go ahead and take a couple seconds to answer questions 1, 2, and 3. All right, and welcome back. And one of the ones I'm going to focus on is going to be number 3. Protein transporters help move molecules like glucose across the cell membrane from high to low. So in this case, This is using our passive. It does not require any energy. Unlike our protein pumps, which are going to require ATP energy, and they're going to be moving against their concentration gradient. So make sure you know the difference between active and passive transport. All right, that is going to conclude our lecture. So again, if you do have any questions, comments, concerns, please feel free to reach out, and I look forward to seeing everybody in the next lecture.